Okay. So I'll start, sir. Yeah.
Yes, sir.
Good evening, everyone. My name is Sangtang. I'm a fourth semester student, MSc Microbiology from Karnataka State Open University. The topic for my presentation is structure, life cycle, and modes of transmission of HIV. So this will be the outline of my presentation: the introduction, structure, life cycle, modes of transmission.
ಶಶಿಕುಮಾರ್ ಮುಗ್ದೆ ಹೋದಲ್ಲ.
Coming to introduction, HIV, we all know
Huh?
it is human immunodeficiency virus, and they are of two types, HIV-1 and HIV-2.
Huh?
And so we are more familiar with HIV-1 because it is more prevalent and highly known for its what to say in fact its disease outcome. We know that it was first isolated in 1983, and it is infecting CD4 expressing immune cell. So what I'm saying is it does not affect all the immune cell, it is affecting only the CD4 expressing cells. So it was a pandemic globally, as known that it is found every part of the world, and in India it was also found first in Chennai in back in 1980s. So, and it is causing it cause acute immunodeficiency syndrome. So acute immunodeficiency syndrome means acute means it's usually by from outside, or there is also autoimmune or genetically immunodeficient syndrome as well. So, but this HIV is causing acquired, which is due to HIV infection. And immunodeficiency means the immune system is low, and it's a syndrome. And so far, there has been no therapeutic cure for HIV, and there is no vaccine available so far. But it can also be controlled. However, it can also be controlled by antiretroviral therapy. Initially, it was hard, and now it is ART, and it can be found in different part of India, supplied by National AIDS Control Organization. Mm. Apart from that, we should also remember that its transmission can be prevented. So, coming to the structure, so this figure, just a second, sir. So this is the structure, how it look like. This is an illustration. So this is an envelope, and these are the proteins which is present outside, and this is the capsid, and then this there are two unique or there are two similar HIV genome, which is unique from other viruses, because most of the virus, I would say all of the virus have individual nucleic acid or individual genome, but this HIV has two similar a pair of similar genome, and there are different protein which is present in itself, they are also in pair. So it is an envelope virus, and it is spherical in shape, and above inside of spherical envelope there is an icosahedral. This is the icosahedral structure, and icosahedral, it is P17 in arrangement. P17, I think this is a very critical thing to understand. For just a few minutes, it may take a whole class. And then the diameter, how big it is 90 nanometer in diameter. So this is the genome structure. I mean, how the genome is coding different proteins. So the left side, 5 untranslated region, and the third prime untranslated region. So the most important thing in this genome is the polymerase, GAG, which is nucleic capsid core protein, and then envelope protein. So in this polymerase, we know that the enzymes like reverse transcriptase, which is very important enzymes for HIV, and then the GAG is the nucleic capsid protein. This is the nucleic capsid protein. These are the nucleic capsid protein, and then the envelope protein as mentioned before, and then the other protein are I'll show it in the next slides. So these proteins, polymerase protein, as I mentioned before, it has protease, reverse transcriptase, integrase, and some other enzymes as well. So here envelope protein, and then polymerase protein. That protein, reverse transcriptase, NAV, VIV, these are the short form, and these are the functions. So uh, unlike the single genome, which I mentioned before, unlike the single genome in other viruses, has a double identical copies, and then the gene provided here are mostly broadly GAG, POL, and POL polymerase is the enzyme which is coding for reverse transcriptase integrase, as I mentioned before, and these three reverse transcriptase integrase proteins, these three are the main thing which is a target site for HIV drugs. I think we know about nucleoside reverse transcriptase inhibitor and non-nucleoside reverse transcriptase inhibitor and integrase inhibitor, and then protease inhibitors that we may take in other classes. So I'll not specifically explain about what are the specific function we can see here, and then precursor polyprotein or viral envelope protein. So the protein is GP160, GP120. So these protein are required for the attachment of the viral protein to viral protein is CD4 cells. And then P55, this is the capsid protein, and reverse pol, it is reverse transcriptase, and then integrase is the enzyme which integrate the genome, HIV genome into the host genome, and protease is the post-translational transduction, or let's say the one which cleave the protein which is produced by the transcription and translation of the HIV protein from the nucleus. And messenger RNA transactivator of apoptosis. This that protein is helping the virus in order to evade apoptosis, and so on. So this is the life cycle, as I mentioned before, and GP120 is the one which is helping in the attachment of the virus, and it is only infecting CD4 cell. And with the help of 6CR4 and CCR5 as a core receptor, the HIV virus binds to the CD4 cells, and then we can see here the HIV virus, the virion, is integrated into the host cells, and after that the reverse transcription takes place. The reverse transcription takes place means this is RNA virus, so the reverse transcriptase enzyme and enzymes transcribe the RNA into DNA. So this DNA is which is further integrated into the host genome, and the host genome here is multiplicating, or it's replicating, transcribing and replicating the viral protein, and these are the new viral protein, and at the same time these viral protein are produced at the same time, and as I mentioned, it is RNA. So this RNA is also the genome itself. The one which are not spliced into the viral protein, it is come as a genome, and then they are integrated into a new integrated into new nucleocapsids, and then the virus is released. And here we found the mature virion, and until this virus is released, even though there is a virion inside, they are not yet infectious. So this is a brief description of that. And then as mentioned before, the binding and release of the virion is by the protease enzymes. So, and coming to the HIV transmission, we all know HIV is mostly transmitted through sexually. In the current genera, this is mostly found. In the early times, it was found to be transmitted through sharing of needles from infected person. However, the first cases of HIV transmission, the HIV infected case, was found in a man who have sex with men. So it's still valid till today that sexual is the main mode of transmission. At the same time, it can also be transmitted through mother to child. So a mother, if a mother is infected with HIV, it can also infect the baby. However, this can be controlled, and there has been many measures in India which have been taken care, and the incidence of HIV transmission from mother to child has decreased. At the same time, blood transfusion is one of the risks, because HIV infected individuals, most of in the early infectious cycle, they don't have any symptoms. So if they have, if they donate blood, the patient who's going to receive the blood will have protection. Therefore, NARCO and National Blood Transfusion Council have taken lots of precaution and tests to detect HIV infection in blood donors. And now we have serological as well as molecular tests to detect HIV infection among blood donors. So this is what I would like to inform. And to summarize, HIV HIV has different protein which is unique. The structure is unique, and the variant is unique from other because it has two identical nucleic acid, which usually no other viruses have. And then the cycle, it is affecting life cycle is that it affect only the CD4 cell, uh, CD4 cell immune cells, CD4 immune cells, and then. Um, as the virus integrates into the host genome, therefore, it is very difficult for the host to detect and to kill the virus in most of the case, except to uh, what to say, to decrease the viral load, or to reduce the viral load. So this is the virus life cycle. And then the mode of transmission, as I mentioned before, it is sexually. It is sharing of needles among the people who inject drugs, or drugs abuser. And then and also, as I mentioned before, it is through blood transfusion and through mother to child as well. And these are my references. Thank you.